Hi, everyone. Welcome back. All right, so I'm Anna Aksakova. I'm a PhD student in the Roth Lab at the University of Toronto, and I'll be moderating this session along with Kevin Kwong, who is our online moderator. And so this session will be shorter because unfortunately Deborah Marks couldn't make it today because she's sick. So instead of having the third talk, we're going to have a longer break. And so it's my pleasure to introduce Alexandra Mandel, who is a PhD candidate from the group of Dr. Guillaume Dis at the Friedrich Miescher Institute for Biomedical Research. So she will be presenting about systematic mutational scanning of the entire human leucine zipper interaction network. Please welcome Alexandra.
Thank you very much for the introduction, and thank you for the opportunity to present my work here today. So every molecular interaction emerges from every molecular function emerges from molecular interaction, because every protein or every molecule that doesn't interact with another one is functionally inert. Even an enzyme that you imagine being there all alone in a reaction tube and not provided with, for example, a substrate or a cofactor that it interacts with cannot have a function. And the specificity of these molecular interactions is encoded in DNA sequence, which means that if we want to go from sequence to molecular function, we need to go via these molecular interactions and their specificities. And in order to understand how molecular functions are perturbed by mutations, we need to understand how mutations perturb molecular interactions and their specificities. Since the launch of AlphaFold, great progress has been made in predicting protein folding and protein interactions from sequence. And although this has, of course, sparked much research and discoveries, AlphaFold is to date neither able to quantitatively predict strength of interactions or how mutations affect folding and interactions. And that's why we in our lab want to develop new experimental and analytical approaches in order to generate large-scale data set of how mutations perturb protein-protein interactions to ultimately create quantitative models to predict protein-protein affinity or specificity from sequence. As a model to study this, I am using the human basic leucine zipper, in short BZIP family, which is a family of protein interaction domains that occur in 54 human transcription factors and can undergo homo and heterodimeric interactions. And they have a relatively simple interface, which makes them perfect as a first model to establish our assays and also learn some first rules of interaction. And there are short alpha helices that are split into this 27 amino acid long DNA binding domain and a 35 amino acid long zipper domain that mediates this interaction by forming coiled coils. And the zipper domain can further be subdivided into these 5 haptids that each consist of 7 amino acids. And these 7 amino acids, or the way they are located in the haptids, are known to be, or to define the general rules of specificity, because each amino acid always takes the same orientation relative to the opposite protein in the dimer, so that amino acids A and D usually form, or always form, the hydrophobic core. E and G can form salt bridges and are generally charged, and B, C, and F are solvent exposed and do not participate in the interaction interface. These general rules of specificity, however, are not that general after all, because they highly depend on context. And if we compare the variability in sequence with the variability in specificity, we see that there is a much higher variability in specificity. And we cannot really predict this from the low variability in sequence based on these simple rules of specificity that we know. Because these rules were actually determined by studying single model BZIPs, such as Foss and June, and didn't look at the entire picture. And therefore, we believe that only by mutating the entire thing, the entire family, we can actually really decipher the sequence-function relationship, including the context, and predict the affinities from sequence alone. And therefore, my project aim was to perform a deep mutational scanning of the entire BZIP network by generating every possible single amino acid mutation in all 54 BZIPs, and then measuring the effect of these mutations on the interaction with their wild type, or with all 54 wild type BZIP partners, so that ultimately for this all-by-all pairwise wild type interaction heat map, I would create one deep mutational scanning assay for each pixel, where I then measure how every possible amino acid at every position in the zipper domain affects the interaction with one of the 54 wild type BZIPs. The assay we're using is Deep PCA, which you already heard about today earlier in Julia's talk. It was developed by Guillaume, my supervisor, during his postdoc time in the LENA lab in Barcelona, and we further optimized this in Basel. It's based on DHFR PCA, this protein complementation assay, where we're using a split enzyme, the DHFR, that we tag, or we use either have to tag one of two proteins that we want to measure the interaction of. And if these proteins interact and the DHFR is reconstituted, it allows growth of cells expressing these tagged proteins in presence of the drug methotrexate, while proteins with non-interacting, cells with non-interacting proteins cannot grow. Our library is that I mentioned before, all possible single amino acid mutations per 54 BZIPs, which has a size of about 38,000. And we pair it with the 54 wild-type BZIPs, which gives us a total of about 2 million combinations that we want to measure the interactions of. And finally, we perform a rather standard deep mutational scanning screen where we express in yeast one pair of BZIPs, variant plus wild-type per cell. We let them grow in this competition screen under methotrexate selection, which results in enrichment of interacting variants. And we can finally infer the strength of interaction from enrichment scores that we measure with next-generation sequencing. However, it is not that easy. We have to take into account that mutations can have different effects on BZIP interaction, and this is also something you already heard before. And here I'm showing you now this model of states that BZIPs can have in the cell. So basically, they can, if they're not bound in a dimer, they are present as these intrinsically disordered monomers, and they only fold upon binding. And this steady-state abundance of monomers is dependent on degradation and synthesis. They can then form either heterodimers or homodimers. The way I presented you now, in our screen, we would only measure heterodimer formation. And this heterodimer formation can be affected either by, of course, a change in free energy in heterodimer formation, but the mutations could also affect this indirectly via changing the rate or the free energies of homodimer formation, and also by changing the steady-state protein abundance, for example, by affecting degradation rate. So actually, to disentangle these different effects, we are also going to measure different phenotypes. We're going to perform different screens. In our case, we're doing we did the heterodimer screen that I mentioned with our mutants that we have as a bait library, and we pair it with the 54 wild type and prey. Secondly, we did a homodimer screen where we, for each mutant in the bait library, combine it with its opposite or with itself on the prey side. And finally, we did two abundance screens, like described earlier, in Julia's talk, where we have for both the bait and the prey mutant libraries, we co-express them with an overexpressed opposite free tag, and then based on random encounters, each BZIP will find each tag on a BZIP will find its partner and reconstitute, and we can then infer the abundance of these BZIP directly from the growth rate of the cell. All right, and with this, I would like to go into a few results that we recently obtained. So these are all quite preliminary, but already, I think, quite exciting, and I'm looking forward also to ideas from your side of what we could do with this kind of data in the future. So this is now the 2 million, or almost 2 million data points that I was talking about before, where we wanted to do this mutational scanning for each pixel in the wild-type heat map. And So here you see this heat map of heat maps where we have in the rows the mutated BZIPs that each have about 700 variants, which gives us a total of about 38,000. And in the columns, we have the wild-type BZIPs. And if we zoom in, these heat maps then look like this, and I will show heat maps like this a few times now, so I will guide you through them. In the columns, you see the positions within the zipper domain, and in the rows, you see the amino acid that was present, and the color then indicates the enrichment score, which in our case is either a log-fold change or a relative log-fold change that are called delta log-fold change, where we calculated how much the mutation altered the interaction relative to the wild-type interaction. So in this case, the DDIT3 mutant score relative to wild-type DDIT3 versus June score. And already with these two different pairwise interactions that I chose, there are some like patterns you can observe. For example, you have some lines going through some column trends where you see at the D and A positions that they often have negative effects. Also down here, or for example, proline and primitive stop codons seem to have negative effects. So I wanted to see if this is the case for all of them or something that is observable throughout the data set. And I calculated average scores for BZIPs that either form heterodimers as wild type or homodimers as wild type. And you can see exactly that the D and A positions have very strong negative effects because these are the hydrophobic core positions, so this makes sense. And the primitive stop codon, of course, disrupts heterodimer or homodimer formation because it truncates the zipper. Also, glycine and proline show a strong negative effect, which makes sense since they are known to be helix breakers. When I compare this to the abundance screens, where I really just measured the abundance of the monomers, this looked quite different, but also matches what we know about BZIPs because they are known to easily aggregate, actually. And if you look at these results, you can see that, for example, here, hydrophobic amino acids like tryptophan, tyrosine, or leucine and phenylalanine, they lower the abundance because they increase hydrophobicity of the zipper and therefore increase the aggregation and thereby proteolytic degradation. On the other hand, here, mutations at the D positions and the A positions stabilize the zipper because these positions in wild type are often hydrophobic. So if we take away the hydrophobicity, we lower the tendency to aggregate. The same is for the primitive stop codon, where this actually also stabilizes the zipper because if we truncate the zipper, we truncate this part that tends to aggregate and the tag that is left isn't as prone for aggregation anymore. I also looked a bit into the effects the mutations have throughout the interactions. And for this one, we only chose those that have at least one significant change in one of the interactions. So theoretically, for each of these 700 mutations that are present in each BZIP, we measure about almost 3,000 interactions. And about half of them show at least one significant change in all of these, in one of the mutations. Yeah. And if you look at this, you can see that almost all of these 1,400 interactions that we measure permutations, that they have both a decreasing and an increasing effect, depending on the interaction where they happen, which strongly shows that we have really this context-dependent effect of what effect a mutation can have. Finally, I wanted to look a bit more into these well-known effects of BZIP mutation, or if our data makes actually sense. And I looked into more of these position-specific effects in the heptad, these core and salt bridge positions, and what effect mutations at these positions would have on interactions, again here, for heterodimer and homodimer forming BZIPs. And I removed the proline, glycine, and stop codons since these are not position dependent. And again, you see that these core mutations are really those that are most disruptive for the dimerization, while the solvent-exposed one are least. And if I compare this to the non-heterodimer or homodimer forming B-zips as wild type, we completely abolish the effects because these don't form dimers, so we don't expect a negative effect from a mutation. Finally, I also compared this, or I looked at this per haptid, and there you can see these are just minor trends, but it looks like haptids 1 and 5 are a bit more robust to mutation, which fits what is known about them, that 4 consecutive haptids should still be enough to form a rather stable dimer, and then the more haptids removed, the less stable it gets. And if we remove the solvent-exposed positions from this, we can further increase this trend. And now, to finish off, I would like to show you 2 specific examples that I think are quite interesting, where we can see how just single mutations are enough to really kind of rewire or like make changes on the network or add something to the network. The first one I picked is CREB5. CREB5 doesn't form a homodimer as a wild type, and it has an asparagine at position 3A. And if we mutate this position 3A, for example, to an isoleucine, we induce a homodimer, a rather strong one. Interestingly, if we compare this to June, which does form a homodimer as wild type and also has an asparagine at this position 3A, here almost every mutation at this position decreases the homodimer. So again, showing this context-dependent effect. If we take this further and we look again at CREB5, we can see that this substitution to an isoleucine only induces the homodimer if it happens in both partners of the dimer. If we look at CREB5 in the heterodimer screen, where we compare all the CREB5 mutants versus wild-type CREB5, this mutation at 3A does not induce a stronger dimerization, showing, if we look at it from an evolutionary standpoint, that a duplication of CREB5 followed by a single point mutation would be able to establish a novel homodimer that's independent of the ancestral CREB5. And we can take this even further and look at all interactors of CREB5. So for wild-type CREB5, the ones that it interacts with are CEBPG and June. And here I plotted now the log-fold changes for CREB5 wild-type versus wild-type partners and CREB5 with this 3AI mutation versus the wild types. And you can see CEBPG and June, which are the strongest interactors here in the wild-type screen, are completely disrupted in the mutation screen. So it loses this ancestral interaction after the mutation, while it gains this homodimer interaction here. And also, these two, BAC1 and BAC2, it barely interacts with or doesn't at all here in the wild-type screen, but upon mutation, we gain these novel interactions, again showing that a single mutation is enough to theoretically produce a novel B-zip that has novel interactions independent of the ancestral ones. The next one I wanted to show you is just a short one to show that these context-dependent effects can also happen at other positions, such as the salt bridge ones. And here I picked JDP2, which usually interacts with many B-zips. Among these, like medium strength interactor is MAF-F. And JDP2 has a glutamic acid at position 2E, a negatively charged one. And MAF-F has that as well at position 2G, which would be the interacting position. And if we mutate this glutamic acid to an arginine at position 2E in JDP2, we induce a dimer here. And this one is positively charged, so this makes sense that we have the stabilizing effect here at the position. Interestingly, if we compare it to MAF-G and MAF-K, which do not interact with JDP2, but also have a glutamic acid at position 2E, the mutation to arginine is not sufficient to induce this novel dimer. So again, this context dependency. All right. And with this, I just showed you some first results that we have. And as you can see, these are just some individual things and trends that we observed, but you may imagine that already with this few individual trends, if we kind of put them together into a simple model, we might be able to predict quite some things, and this might make sense in a combined together. And this is what we will work on now, and also this thermodynamic model that I mentioned before, to detangle the mutant effects and really pin down what each mutation does. But where it might get a bit tricky is this context dependency of the mutations. And this is why, in parallel, we're also working with another group at FMI that's led by Friedemann Zenke, and the major work is done by Bastian, a PhD student. And they're training a neural network as a model of these sequence-function relationships in BZIPs. And they're using a transformer model, which should be very well suited to learn these context dependencies, because they're also used for natural language processing where they do that. And we then want to really predict these specificities from sequence alone, and also use this neural network to predict novel BZIPs as potential tools for synthetic biology approaches, or also competitive inhibitors with a pharmacological aspect, which would be really interesting. And with that, I reach the end. Thank you so much for this opportunity today. And also, if you want to look at it again, I have a poster on Thursday. Please check that out. And I want to thank my group, and I think some of them are watching now and even stayed late in the lab, so hi in Basel. And I wanted to specifically mention also our computational platform with Charlotte and Michael, who developed this great analysis platform for us, an analysis package that we will also share soon, and I hope will also help a lot of you with your work. So thanks.
Hi, really nice talk, an impressive scale. I have maybe two-part question. The first is something I think Fritz a long time ago brought up with me on protein complementation assays versus other interaction profiling assays, that they tend to measure more K-on than KD, in the sense that once they come on, they come sometimes hard to come off. So I'm wondering if you saw any of that in your assay, and we've seen some of that in a different split complementation assay. And then the second part is the DNA binding domain in BZIPs have also been shown to confer specificity, both through the binding of DNA causing differences. And I'm wondering if you can comment on those factors as well.
Yeah, so maybe the second one first, because I didn't get the first one completely. You might have to repeat that. So we still have the DNA binding domains on there, so we're basically excluding this mostly right now to look at this. Of course, we're expressing human BZIPs in yeast, so we don't know what effect the DNA binding has there. We know that they are in the nucleus in the yeast cells, so they might actually bind to DNA. But we also already did some confirmation experiments, small scale in mammalian cells that are done by my colleague Roman. And it looks like we can reproduce a lot of the stuff that we see also in mammalian cells, so it looks quite promising, yeah. And can you say the first question again, please?
Yeah, we ran into this a little bit where when we were looking at human BZIPs through a split complementation assay, that was a split adenylate cyclase system. We got a lot of discordance with like Amy Keating's work on measuring KD, or where like differences between the in vitro assay, at least in binding measurements. And I think it was largely because it was, I'm guessing because of the avidity of the remaining split protein. So in your case, the split DHFR. So I'm curious if you saw some of that thresholding effects or some of the known mutations that are thought to affect K-off over K-on.
Yeah, not yet. We haven't looked that deeply, I would say. I am trying kind of to verify this to some in vitro data that is published already, but so far I haven't done a lot there, so I cannot comment a lot on that yet.
Maybe, do you see a relationship between KD and your signal? Like
We
the
have-
stronger signal looked, yeah.
I see, if I compare it to, I mean, we don't have transformed this into KDs or anything yet, but if I compare it to like this in vitro paper from 2013, there was one from Amy Keating's lab, it's kind of, there's a little bit of a correlation there, but I haven't looked in great detail yet, yeah.
We have a question from the online audience.
Mm-hmm.
So thank you for an impressive talk. Do you discriminate between aggregation and degradation in your abundance assay?
No, we just measure abundance, but like only, yeah, if they're there or not, but we don't look into what's specifically happening, yeah.
Awesome, thank you.
All right, thanks.